Hi, it's Ileana here, and I'm very much interested in genetics and also in researching what nanomedicine and nanodevices is. That's what I did on the planetary corporations on Mars. So I'm just looking for information on Earth in regards to what is going on with nanomedicine, nanotechnology on Earth. There seems to be a movement towards nanomedicine and stuff. So what I found recently is this living computer's RNA circuits transform cells into nanodevices. This is what's the latest that I found on nanodevices, because that's what was a big part of my work on the Planetary Corporation Cybernetics Labs on Mars. So I thought I would cover this article today. So the title is ASU, Biodesign Researcher Shows How Cells Can Be Induced to Carry Out Computations with Implications in Health and Energy. The interdisciplinary nexus of biology and engineering, known as synthetic biology, is growing at a rapid pace, opening new vistas that could scarcely be imagined a short time ago. In new research, Alex Green, an assistant professor at ASU's Biodesign Institute, demonstrates how living cells can be induced to carry out computations in the manner of tiny robots or computers. The results of the new study have significant implications for intelligent drug design and smart drug delivery, what I call biological drug serums. That's what was done on the Planetary Corporation Cybernetics Labs. Also green energy production, low-cost diagnostic technologies, and even the development of futuristic nanomachines capable of hunting down cancer cells or switching off apparent genes. That's very interesting because we also did gene manipulation, and nanomedicine is very much involved in doing that. And basically it's regeneration medicine as well. We're using very predictable and programmable RNA/RNA interactions to define what these circuits can do, Green said. That means we can use computer software to design RNA sequences that behave the way we want them to in a cell. It makes the design process a lot faster. So they say that the study appears in the Advanced Online edition of the journal Nature. Designer RNA. The approach described uses circuits composed of ribonucleic acid or RNA. These circuit designs, which resemble conventional electronic circuits, self-assemble in bacterial cells, allowing them to sense incoming messages and respond to them by producing a particular computational output, in this case, a protein. In the new study, specialized circuits, known as logic gates, were designed in the lab, then incorporated into living cells. The tiny circuit switches are tripped when messages, in the form of RNA fragments, attach themselves to their complementary RNA sequences in the cellular circuit, activating the logic gate and producing the desired output. The RNA switches can be combined in various ways to produce more complex logic gates, capable of evaluating and responding to multiple inputs, just as a simple computer may take several variables and perform sequential operations like addition and subtraction in order to reach a final result. The new study dramatically improves the ease with which cellular computing may be carried out. The RNA-only approach to producing cellular nanodevices is a significant advance as earlier efforts required the use of complex intermediaries like proteins. Now, the necessary ribocomputing parts can be readily designed on a computer. The simple base pairing properties of RNA's 4 nucleotide letters, A, C, G, and U, ensure the predictable self-assembly and functioning of these parts within a living cell. Green's work in this area began at the Weiss Institute at Harvard, when he helped develop the central component used in the cellular circuits known as an RNA toehold switch. The work was carried out while Green was a postdoc working with nanotechnology expert Peng Yin, along with the synthetic biologists James Collins and Pamela Silver, who are all co-authors on the new paper. The first experiments were in 2012, Green said. Basically, the toehold switches performed so well that we wanted to find a way to best exploit them for cellular applications. After arriving at ASU, Green's first grad student, Duo Ma, worked on experiments at the Biodesign Institute, while postdoctoral scholar Zhongming Kim continued similar work at the Weiss Institute. Both are also co-authors of the new study. Nature's Pentium chip. The possibility of using DNA and RNA, the molecules of life, to perform computer-like computations was first demonstrated in 1994 by Leonard Adelman of the University of Southern California. Southern California, interesting. So a lot of stuff coming out of that. Since then, rapid progress has advanced the field considerably, and recently such molecular computing has been accomplished within living cells. Bacterial cells are usually employed for this purpose as they are simpler and easier to manipulate, or also mitochondria, membranes and the cells within them that can be also used in applications with RNA and nanomedicine. The technique described in the new paper takes advantage of the fact that RNA, unlike DNA, is single-stranded when it is produced in cells. This allows researchers to design RNA circuits that can be activated when a complementary RNA strand binds with an exposed RNA sequence in the designed circuit. This binding of complementary strands is regular and predictable, with A nucleotides always pairing with U and C always pairing with G. With all the processing elements of the circuit made using RNA, which can take on an astronomical number of potential sequences, the real power of the newly described method lies in its ability to perform many operations at the same time. This capacity for parallel processing permits faster and more sophisticated computation while making efficient use of the limited resources of the cell. Logical results. In the new study, logic gates known as AND and NOT were designed. An AND gate produces an output in the cell only when two RNA messages A and B are present. An OR gate responds to either A or B, while a NOT gate will block output of a given RNA input is present, if a given RNA input is present. Combining these gates can produce complex logic capable of responding to multiple inputs. Using RNA toehold switches, the researchers produced the first ribocomputing devices capable of 4-input AND, 6-input OR, and a 12-input device able to carry out a complex combination of AND, OR, and NOT logic known as the disjunctive normal form expression. So I think nanomedicine, nanotechnology are merging together with human biology. And this is the result of using RNA in cells to target disease, to remove it, and to also use nanotech as a driving force to deliver medical applications. So it's basically going to be for medical applications to deliver nanomedicine to cells that can then target the disease with the RNA to remove it, like cancer. When the logic gate encounters the correct RNA binding sequences leading to activation, a toe-hold switch opens and the process of translation to protein takes place. All of these circuit sensing and output functions can be integrated into the same molecule, making the systems compact and easier to implement in a cell. The research represents the next phase of ongoing work using the highly versatile RNA toe-hold switches. In earlier work, Green and his colleagues demonstrated that an inexpensive paper-based array of RNA toe-hold switches could act as a highly accurate platform for diagnosing the Zika virus. Detection of viral RNA by the array activated the toe-hold switches, triggering production of a protein, which registered as a color change on the array. So this Nanodevices technology combined with biology can also detect viruses. That's interesting. The basic principle of using RNA-based devices to regulate protein production can be applied to virtually any RNA input, ushering in a new generation of accurate, low-cost diagnostics for a broad range of diseases. The cell-free approach is particularly well suited for emerging threats and during disease outbreaks in the developing world where medical resources and personnel may be limited. So basically, the nanomedicine passports might be coming to us in the near future, where with nanoparticles you get antibodies for immunity of this or that from diseases, viruses, etc. Or you get a disease removed from your body, like cancer, it's evaporated with nanoparticle technology delivered through the RNA inputs. The computer within. According to Green, the next stage of research will focus on the use of the RNA toe-hold technology to produce so-called neural networks within living cells, circuits capable of analyzing a range of excitatory and inhibitory inputs, averaging them and producing an output once a particular threshold of activity is reached, much the way a neuron averages incoming signals from other neurons. Ultimately, researchers hope to induce cells to communicate with one another via programmable molecular signals, forming a truly interactive brain-like network. Because we're using RNA, a universal molecule of life, we know these interactions can also work in other cells. So our method provides a general strategy that could be ported to other organisms, Green said, alluding to a future in which human cells become fully programmable entities with extensive biological capabilities. So this article really discusses the ins and outs and nuts and bolts of futuristic nanomedicine, nanotechnology applications. So I thought I would cover the article because this is happening now. These studies are happening now. And this article was actually released in July 26th, 2017. So the article is a few years old. But this technology of nanomedicine, nanotech, being fused with biology, it's already being studied and it's being close to being implemented in our near future. So that's something to be aware of. I am not a proponent of transhumanism and merging nanotechnology with biology, but there's some very tiered applications to this and repercussions, and if nanomedicine is used right, it can remove diseases, and it could stop diseases, and it could stop viruses. So it could be implemented for the good or for the evil to produce stuff, viruses, diseases. You can use it for positivity or negativity. It all depends how the nanoparticles are programmed to begin with. What's the purpose of them? What are they being used for? So that's something to think about. Again, I'm not a proponent of transhumanism or negative timelines or creating negative futures. I'm just reporting on what's going on in this world. What is happening? What is being processed and potentially implemented in the near future? Awareness brings change. Awareness can bring positivity, or it can bring negativity. It all depends on how you see things in reality and the polarity of energy, what comes and goes. So how you see and interpret things is very important. This is just an article telling us what RNA circuits can transform cells and do with nanodevices. So this is an article, but this is being studied. This is already being trialed as well. So it's something to think about and understand. Where are we going with this? Why is this happening? What will it bring to us? Do we want this or do we not want this? Do we need this? So that's something to think about. So thank you so much.